Hello. We're going to talk about polygenic risk scores and specifically the challenges that they face in terms of implementation, both at the technological level, the technical level, as well as alluding to the issues that face the implementation of polygenic risk scores in terms of the regulatory and the business model considerations, but really focusing on the technical issues. So the challenges for the application of polygenic risk scores is that at present, because they are based on existing data sets of disease, they are really only applicable to individuals of European origin. And that's because most of genetic studies that have been carried out to date, genome-wide association studies, the foundation for polygenic risk scores, almost all of the large-scale GWAS studies have been carried out in populations of European origin. So the solution for that, of course, is ultimately to carry out large GWAS studies in other population groups. And then in terms of the utility for the patient and for the healthcare delivery system, really that's an unexplored area at this point. 3 value cases have been highlighted, but over 1,500 trait polygenic risk scores have been generated. And so really what the present situation is that we're faced with challenges that we can see glimmers of how they can be solved, or we can see clear roads to being solved, but we're still in that window of time where we can see the potential applications of polygenic risk scores. We know the problems we need to address and solve, and so there's a window of opportunity to get everything right. And then just in terms of the regulatory and the business model, there is not a valid validation that's been done for the payer and the regulatory approval. And that's going to require investment by someone with a business model. So there's need for innovation there as well in terms of exactly how polygenic risk scores will be implemented into the US healthcare system. But focusing on the technical issues and taking a step back, the goals of human genetics are, from my formulation, to predict disease risk, and that's of benefit for population risk prediction, for health resource planning and allocation, for individual risk prediction, so enabling treatment, patient management, treatment, and lifestyle decisions. And then finally, therapeutic intervention, so identifying reversible processes that are driving disease progression. So we're going to focus on, that top part and most, for the most part, on individual risk prediction, because I think that's where that's the appropriate place to think about polygenic risk scores and how they, what the challenges are and what are the benefits. So the genetic, really the thinking, the development of polygenic risk scores is based on some kind of basic characteristics of the genetic architecture of disease. So early on in genetics, in human genetics, I should say, there was a focus on a few loci of large effects. That was what we knew how to identify, how to study. So Mendelian diseases, recessive disease, dominant disease of large effect, where really could be mapped to single loci. On the flip side, starting more than 10 years ago, it's really starting with the Wellcome Trust Case Control Consortium. It was identified that it was possible to effectively measure and identify many loci of small effect, so polygenic. So this is what I'm referring to as the genetic architecture of disease, these two extremes in terms of genetic effects. And polygenic risk scores are really built on those many loci of small effect. So how do we measure those and use those many loci of small effect to provide a risk prediction for an individual? So in the earliest days of the genome-wide association studies, people began to see if they could add together the significant loci that were identified for any particular disease. So this is not in the very early days, this example that I'm giving here, but it illustrates the same basic point. So this is from a paper in Circulation, where a polygenic risk score based on 57 SNPs, so 57 loci, was created. So again, looking at the allelic status at each one of these loci for an individual allows one to add together the effects, and the effects do appear to be additive, add together the effects and produce an aggregate risk prediction, the aggregate contribution of each of these 57 loci. So there's a couple of points. One is early efforts focused on loci that had met on their own genome-wide significance, so an arbitrary p-value threshold. And that put a limitation on the number of loci just because the sample size of the case control studies was early on relatively small. So the earliest efforts at polygenic risk scores were based on 5, 10, et cetera, loci that had met genome-wide significance. And it did provide risk prediction, but not at a level that was useful for predicting risk for an individual, for making an individual decision as to treatment or intervention. So that brings us to the second point, which is shown in the illustrated, I should say, in the bottom right, and that is that individuals, if we really focus on the red bar graphs, individuals at high genetic risk because of they carried the risk loci for many of these 57 SNPs, they respond well to statins in terms of the reduction in cardiovascular risk. So that's illustrated by the fact that the hazard ratio there, about 0.56 of the individuals on statin versus those on placebo. So that's, again, an important point. These 57 SNPs, when used together to create a polygenic risk score, led to information that was meaningful for treatment intervention. So another advance which has happened most recently is that it was recognized that the limitation of only using variants, loci that had met genome-wide significance was somewhat arbitrary because doing a larger study would bring more loci into that significant range. And so approaches were developed for using many variants. So in this case, there's what's called a genome-wide polygenic risk score, which uses over 6 million variants to construct a polygenic risk score. So as you add in more variants of smaller effect, of course, each individual one has less and less impact on the overall score. But again, you're sampling from a much larger set. And again, you can see that, that there is a clear segregation in risk, as shown there in terms of the polygenic risk score percentile, the case control. But the other thing is because one has so many variants, one can really start subdividing the risk into smaller and smaller percentiles or bins of risk. So obviously with 6 million variants, one is not limited in the number of categories that one can place individuals in terms of risk. And what that showed us was that by using more and more risk variants and being able to look at, in this case, the top 1 percentile is illustrated here, that one could develop polygenic risk scores for certain complex disease that had predictive power equivalent to single loss of functional alleles. So the comparison here would be for coronary artery disease, this 5-fold increased risk of the top 1 percentile of risk compared to all the other individuals, so that nearly 5 to 1 is similar to what the increased risk that occurs with variants that cause familial hypercholesterolemia. So similarly, for a number of other disease conditions, you can see that the individuals at the highest risk have risk which is many multiples of what the general population risk is. So what is the implications of that? So again, using sticking with the example of cardiovascular risk, which is I think where we have the clearest picture of the potential utility of polygenic risk scores. There's a standard approach, the pooled cohort equation approach, where one uses a number of different clinical factors. This illustrated here is cholesterol, smoking, blood pressure, also included are diabetes status, etc. Gives you a relative risk prediction. So polygenic risk can also give you a relative risk prediction. And importantly, what has been recognized is that the risk of the pooled cohort equation factors is independent of that of the polygenic risk. So in other words, polygenic risk gives you another powerful indicator of cardiovascular risk. So then that allows then a combined risk to be potentially calculated, where the clinical risk and the polygenic risk are combined together to give different risk categories. So that gives us an idea of how polygenic risk factors can be applied in the clinic. As I said, cardiovascular risk is something where we are used to thinking about primary prevention, secondary prevention, so managing the clinical situation, the clinical factors with medication and lifestyle changes to avoid an outcome, in that case, a cardiovascular event. In many other disease situations, the ability to predict risk has not been robust, and therefore the paradigms, the clinical management approaches for managing risk have not been developed to that degree. But you can really see from that how what the potential possibilities are. So I moved on to the next slide a little bit prematurely, but the point is that is to illustrate one of the greatest barriers to application of polygenic risk clinically right now, and that is that the data sets that we have for constructing these risk scores are based on European populations. It's very graphically illustrated here. You can see the big fat red segment of the curve going across in the middle there. That's the number of individuals who have been included in genome-wide association studies. There's colored bars. There's a rainbow there at the bottom of other populations. I'm going to be telling you about the purple line there, where there's very, very few individuals have been included in genome-wide association studies, that is South Asians. And then on the right-hand side is a reminder of the relative proportion of the global population of the different groups there. And you can see that South Asians make up a very significant fraction of the global population. So there's a very significant opportunity to introduce polygenic risk prediction into patient management for South Asians. So I'm going to skip past that really quickly. So the question then is, we can recognize this disparity. How do we solve it? I'm going to be telling you a little bit about studies that I've been involved in that have sought to address that deficit. It's based on a consortium, the Genome Asia 100,000, 100K. So the first phase of this was published last December in Nature. I'm not going to spend any time talking about that, but tell you about the overall approach. So that what was published there last December, I'm going to call phase 1, and that was the first part of the efforts to address this disparity, where we used whole genome sequencing of genomes from all across Asia. Asia is a very large and diverse part of the world, of course. That helped us develop strategies for more regionally focused efforts. And what I'll tell you a little bit about is phase 2, where, or one of the phase 2 activities focused on India. So again, using whole genome sequencing to build a description of the population structure, and then tools that will allow genetic studies to be carried out more effectively and economically. So optimized reference panel and optimized SNP array. And then ultimately what that does is make it possible to generate large data sets, case control and other data sets for disease that occurs in South Asians. It allows us to have a much better prediction of genetic risk in South Asian populations. So South Asia has very distinct origins from European populations. That's shown on the left-hand side. I won't spend any time talking about that. That's referring to population history from tens of thousands of years ago. So that's the top bullet points across the top, that South Asian origins are very different from Europeans or East Asians, hence the failure of European genetic data to adequately predict risk in South Asians. There's many, many thousands of population groups. There are also strong founder effects which occur. And what that implies in terms of genetics is that functional variants, missense, and loss of function variants occur at higher frequency. And then finally, there are much higher rates of endogamy and consanguinity. So in other words, the chances are that one's parents are more closely related than is common in Europe occurs at a higher rate in South Asia. So importantly, what we did was we focused on data that came from individuals who were in the healthcare delivery system. So in other words, individuals who would be sampled in medical genetic studies. And also importantly, given the diversity of humanity across South Asia, we focused on the three corners of the subcontinent as capturing the genetic diversity across the subcontinent. So that's shown there in the inset map on the left-hand side, where we sampled from West Bengal and Bangladesh, from South India, and from Pakistan. So we used high coverage whole genome sequencing to sample that diversity. You can see in this UMAP plot that's in the middle part, which captures the effects of genetic diversity by clustering together individuals whose genetic variation is limited between them. So people who are genetically similar, and spreads apart individuals who are more dissimilar. So you can see that there are the three geographic regions, the individuals cluster together, and that they cluster separately. And you can see, of course, a rough map of the subcontinent that is formed. We can recreate the corners of the geography in the genetic diversity. And then importantly, there is a lot of substructure. So in other words, clustering together and clustering apart within each of those regions. And we can get a picture of what the basis is for that by using some of the data from our first phase of our project, where you can see that there's colored dots overlaying the gray dots of the general population. And you can see that the colored dots, which are individuals from identified groups, illustrate what some of those patterns are in the overall general population. From that data, what we did was we generated a reference panel. So a reference panel allows one to impute missing genotypes. So a foundation of the GWAS era was that relatively sparse genotyping arrays could be used to capture a much larger fraction of the total variation, essentially by interpolating between genotypes. And a reference panel allows one to interpolate effectively. And so what has been a standard is the 1,000 genomes reference panel. That's shown here with the red or orange dots. And then the blue dots are shown the output from using the reference panel that we created from those the large number of South Asian individuals. And what's shown on the x-axis is the allele frequency. On the y-axis is the accuracy, the r-squared for each genotype, where we interpolate, and then we look at the accuracy of those interpolations, those imputations. And you can see that 1,000 genomes never gets up above about 0.8. No matter how common the allele is. So even at a 50.5 minor allele frequency of 0.5, so it's a 50-50 guess, still it doesn't do tremendously well, whereas there's a significant improvement with the Genome Asia phase 2 panel. So what that then implies is that much more accurate genotyping can be done using SNP arrays with the use of this reference panel. And then what we also did with that data set of individuals from across South Asia is we generated a new genotyping array. So again, it's relatively sparse compared to the overall amount of variation in the genome, but by combining this new genotyping array together with the reference panel, we can create very dramatic improvements in the ability to genotype South Asians at a cost which is much lower than using high coverage whole genome sequencing. So what's illustrated here is the number of variants that are directly genotyped, the number of variants that affect protein coding capacity on the new array on the left-hand side versus a standard array, the Illumina GSA array. And you can see that we are genotyping many more functional variants in the genome with this new array. So that combined with the improved imputation of the reference panel produces a much dramatic improvements in ability to carry out large-scale data sets, which will then allow much improved polygenic risk scores. So the phase 3 I had on the overall strategy, and this is what is meant by phase 3, which is really combining these tools together with high quality patient population data sets, sample and database, clinical database sets to create a very large clinical genomic databases that will allow effective and accurate, will allow economically the creation of very large-scale data sets. So I will stop there. There's many individuals who have contributed to all of the work, everything I've said. I tried to have citations on the slide for the literature citations, but I have many collaborators that have contributed to the work I summarized at the end. Thank you.